So hi everybody, my name is Beata, as Marlene has said. I'm going to talk to you about a novel idea which we have been working on with Frederick Thomas and Robert Gatenby, and I should have put on the sign that in progress we are actually just developing these ideas. But before I go into the details, I just thought to give you a background to those in the audience who are actually not from cancer research. So probably all of you know that cancer is not one disease, it's actually a large complex of different diseases. And every cancer behave actually differently depending on the origin of the cell type. Cancer has been called the disease of the cell or the disease of the genome, but actually what's known and what's similar in all cancers, that it starts because abnormal cell growth goes out of control. And cancer can result when control over cell growth is lost. So as Amy has showed you, cancer is not unique to humans. It occurs in every animal nearly in the planet. And as again we see from Amy's study, animals are actually an excellent model system to study cancer. So what causes cancer? Majority of cancers are caused by environmental factors. A smaller proportion of cancers are inherited, and approximately 15% of cancers originate from infectious etiologies. However, today I would like to talk to you about cancers which are relatively rare. These are cancers which don't have any infectious underlying factors, but they are contagious. They spread from animal to animal or human to human. So contagious cancers are actually frequent or relatively frequent in humans. Approximately 3,500 women develop cancer during pregnancy, and rare cases of mother to fetus and fetus to fetus transmission occurs. Prior to the study of PEN in 1978, there was no selective screening or systematic screening for cancer histories in organ donors. So approximately 1/3 of organ recipients develop cancers. Since the screening has been in place, the number of cancer cases dropped in organ recipients. And there are also a couple of cases, well-known cases in the literature when, without any underlying factor, human-to-human transmission occurred. Probably the most well-known case is when a surgeon contracted a malignant fibrous histiocytoma from a patient during surgery. So these are just a few examples. But I thought to talk to you today about the most well-known cases of transmissible cancers in the literature, and they originate from animals. So one of the most well-known cases is probably the Syrian hamster case, which in the 1980s there was a laboratory population when cancer spread from animal to animal via mosquitoes, and the animals were kept in different cages. But probably the most well-known cases are the canine transmissible venereal tumor disease and the devil facial tumor disease. And I'm going to focus on these two diseases today. So I just thought to give you a quick background about them, just to tell you what they are if you are not familiar with the literature. So CTVT appeared approximately 11,000 years ago, while the FTD is a much younger cancer. The first sightings were in 1996. Both cancers, transmissible cancers, emerge due to low genetic diversity of the host species, and both of them are infectious and direct contact is required for the infection. Both cancer requires physical interaction between the two host individuals. CTVT spreads during sexual intercourse, while the FTD spreads during biting. Interestingly, CTVT is rarely fatal, while the FTD is nearly always fatal. In CTVT, we see a spontaneous regression within 6 to 9 months of the appearance of the first lesions, while the FTD hardly shows any signs of regressions. CTVT rarely metastasizes, while the FTD is a highly metastatic tumor. And as you can see, CTVT spread across the globe during the last 500 to 400 years, and the FTD is only restricted to the island state of Tasmania, south of Australia. So as I showed you, there are not that many transmissible cancers in the wild. And if you think about that a cell which is able to proliferate unlimitedly and which is able to transmit from host to host, should be selected for, should have the highest fitness, there should be much more transmissible cancers occurring in the wild. So why are they not more common, and how do they evolve? So first I will tell you about how do they evolve, and then I will show you an idea which we came up with Fred and Bob Gettenby. But before I show you about the evolution of transmissible cancers, I just step back for 1 second, and I don't have to go into details. Carlo gave a really good background and good review and introduction about the evolution of cancers. One main point which I would like to make is, as we know, cancer is a disease of a clonal evolution within an individual. So if a subclone, a mutant subclone acquires high proliferative rates, it's able to diffuse and spread to the novel tissue microecosystems. But the ultimate fate of this tumor or of cancers is that they actually die with the death of the host. So the difference between non-communicable, non-transmissible cancers and transmissible cancers, the transmissible cancers are actually the disease. A cancer is a disease of clonal evolution across individuals. So in this system, the cancer cell, which was able to invade new tissue microecosystem, is actually able to not only adapt to the microecosystem of its individual host, but it's able to spread from host to host. So it's actually able to survive infinitely. Okay, so I promised to tell you about the evolution of transmissible cancers. So as I told you already, CTVT is the oldest naturally occurring cancer lineage or cell lineage in the wild. It's over 10,000 years old, while DFTD is a relatively younger cell lineage. Both of them infected over 100,000 of the host species, dogs in the case of CTVT, and 100,000 devils in the case of DFTD. Both of them shows the signs of chromotripsis, so both of them most likely have developed due to or via a catastrophic chromosomal failure and chromosomal explosion. What's interesting that the genome is relatively stable in both cancers. While CTVT chromosomally stable, there are no karyotypic changes in CTVT anymore. The FTD is still evolving. It still shows chromosomal rearrangements. Several strains have been described lately, and we know that due to selection pressure, the cancer actually can increase its chromosomal numbers. I couldn't find any information about telomere homeostasis in CTVT, but we know that the FTD is actually dynamically changes its telomere length over time. The CTVT transcriptome shows upregulated autoimmune responses, and we were quite surprised not to see any strain-specific difference at the transcriptome level of the FTD. Epigenetic regulation is relatively stable in CTVT, while in the FTD, epigenetic regulations are undergoing dynamically over time, and we see a dynamic change in the different cancers. So what's interesting in CTVT that it has two stages of progression. And the first stage is when it infects the new host, the new dog. And during that progression stage, MHC is not regulated on the cell surface, hence it can avoid immune recognition. But during the regression stage, MHC is upregulated again, and the immune system of the dogs can recognize the cancer. Unfortunately, in the FTD, MHC is only down-regulated on the surface, hence the cancer can avoid immune recognition and can spread from devil to devil. So what does this all mean? So to us, it meant that CTVT has adapted to its niche. It has reached an evolutionary equilibrium in its micro and macro environment, meaning within its host and also between the different hosts in the individuals, while the DFTD still shows the stepwise evolutionary steps and smaller, rapid minor pheno and genotypic alterations. So it's still evolving. So by looking at the two cancers, we can see the end product of cancer evolution and the middle stages, the stepwise processes of cancer evolution. If I come back to my big question that why transmissible cancers are not more common. And we came up with the idea to try to understand cancer evolution by using an example of invasive species. And I decided to show you an Australian example since I come from Australia. And I try to convince you that the example of invasive species can be applied to cancer, transmissible cancer evolution. So what makes a species a successful invader? So in 1935, 102 cane toads were introduced into the Queensland region of Australia. And during the last couple of years, they have spread across the tropical regions of Australia. So what are the key factors to become successive and to become a successive invasive species? One of them is the number of animals introduced into the new area. The numbers have to be over a threshold for the species to be able to spread. The presence of vectors. In the case of cane toads, they were humans. The possibility for high proliferation and high reproductive potential. And the presence of a permissive habitat, meaning that the habitat of the novel habitat is usually quite similar to the habitat of origin. We know from invasive species, successful invasive species, that they change their microenvironment or their novel environment where they were introduced. They reduce competition and they reduce predation. So if I use the same analogy for transmissible cancers, it's the same that we need to have an enough amount of cell lines or cells to be transmitted, to be successfully transmitted. The presence of vector is a key factor. So as I told you, both in the devil and the canine tumor, the vector was the physical contact between the hosts, between the animals. We know cancer cells have high proliferation levels, so they are able to outcompete the novel cells, surrounding cells. And the presence of permissive habitat in both cancers, we know that they originated from highly bottleneck species with low genetic diversity, which facilitate the cancer transmission by evading immune recognition. We know from cancers that they change their microhabitat, they increase acidity and hypoxia in the surrounding tissue, and hence reducing competition, and they reduce immune recognition or predation by downregulating MHC on their cell surface. So we propose that a perfect storm is necessary for a transmissible cancer to evolve or to develop. As you know, both at organismal and ecosystem levels, very few species and cells become invasive. The successful transmission of cancer requires a perfect storm with the rare confluence of multiple host and tumor cells traits to be present at the same time. What are these traits? So from the species side or cell side, the key factors are fast growth, rapid reproduction, high phenotypic plasticity, ecological tolerance, and being generalist, and also being able to associate with vectors. From the ecosystem side, research-rich, disturbed environments open up empty niches or novel niches. The site of origin and the site of end of transmission should be quite similar. Presence of reduced predation, competition, and again the presence of vectors. So finally, I just would like to acknowledge all the people with who I've been working during the last couple of years and months, funding. And if you are interested in our research, please visit our website. Thank you very much.